So today I will talk about cortical dynamics in gait adaptation and how these can be investigated in single trial using EEG source imaging. So if we think about gait research, it's often concentrating on the motor control of steady state walking, meaning that people study the neural control of movement execution of steps and the muscle control of walking. However, this is an incomplete model of gait. So when we think about navigating in our everyday life, it becomes kind of obvious that walking is a cognitive task. So let's take this. A second, let's watch this video for a second. So let's imagine walking on a busy street with a lot of other pedestrians. So this is an example of the of a crossing in Tokyo, which is one of the busiest intersections in the world. So there are 6 or 7 different streets intersecting at this corner, and people can cross to any corner. So now imagine walking across this intersection. It's a highly demanding task because you have to constantly avoid other pedestrians, adjust your gait, recognize where you have to go, and predict where other people are gonna go. So let's watch it from the top for a second. Yeah, so this is a highly demanding task. So let's go back to the presentation. So I think we can agree that gait adaptation requires cognitive control. Here on the picture, we can see a little girl mastering an obstacle course, but it's not so clear which control mechanism actually come into play when that little girl has to adapt her steps to step over those obstacles. So one possibility is that adjusting a movement may require inhibition of the ongoing or planned movement. So for example, let's take the example, if you're at home and your cat runs into your path while you approach your kitchen, you need to quickly stop your ongoing step and find an alternative foot landing position before you fall down. So why is this important? So why do we actually want to study the neural mechanisms of gait adaptation? Because there are a lot of gait impairments in elderly and neurologic populations, for example, Parkinson's, that have gait adjustment deficits, and these deficits lead to fall and related injuries. However, it's not clear what are the mechanisms of these deficits. So gait impairments in general in Parkinson's and the elderly have been shown to be related to a loss in executive functions. And some studies have shown that actually walking and stepping over an obstacle require top-down inhibitory control. So, for example, in elderly, attention and executive function tests that measure response inhibition predicted the risk of falls 5 years later. And it has also been shown that elderly have a decreased ability to implement top-down inhibitory control during obstacle stepping in a dual task walking, where participants have to do a secondary task that requires stopping a movement. So the brain that you can see here on the slide is from a structured review. And this review found that there is strong evidence for Parkinson's and elderly people that the prefrontal cortex is involved in these gait impairments. However, when we want to study the neural control of gait adaptation, we need to consider that there also may be different strategies for obstacle avoidance, and these different strategies may require different cortical resources. So for example, when we approach an obstacle, we can either step over it by lengthening our step, or we can step before it by shortening our steps. So step lengthening usually requires an increase in propulsive forces, while step shortening requires the active stopping of the swing phase of the leg. So it may actually require response inhibition. So here in this study that I will show, we will look at EEG signatures of inhibitory control that have been previously observed in seated button press tasks. So on the left, you can see that there is a beta power increase in right frontal cortex, and this has been shown in seated button press tasks that require to stop an ongoing movement. And these beta power increases have been shown to be part of a prefrontal basal ganglia stopping network. On the right, you can see error-related potentials, which occur after an erroneous action, for example, a wrong button press, or 250 milliseconds after negative feedback to a wrong action. And these error-related potentials have been shown to be part of a performance monitoring network that basically tells us when we have to adjust our behavior. I cannot, okay. So we will investigate these EEG signatures of inhibitory control in a gait adaptation task. So I will first talk about the experimental paradigm, then about the behavioral results and the methods, and then I will talk about the analysis and results of event-related oscillations, and then about single trial analysis of the error-related potentials. So here we can see the paradigm. So there's a participant walking on the treadmill with auditory cues. The task was to adapt his steps to the cues. So first the cues were delivered with his preferred cadence, so the cadence that he was comfortably walking with. And then the cues got faster or slower, and participants had to adapt their steps to the new cue tempo. So the speed of the treadmill stayed always the same during the experiment. So basically participants had to adapt their steps by making larger or longer or shorter steps. So we can see that now he's shortening his steps. Now he's going back to his preferred cadence. And then now he's switching to lengthening his steps. So we recorded 108 EEG electrodes. We had in-ear headphones deliver the auditory cues, and we measured heel strikes using foot switches at the heels of participants. This is again a schema of one trial. So participants first walked 10 seconds without cues. Then the cues were delivered at their preferred cadence, and then the cues got faster or slower, and they had to adapt to the new cue tempo. The preferred cadence cues, they were computed during every trial, so during uncued walking, to make sure that if there are small variabilities in walking cadence, that participants always get delivered the cues at their preferred cadence. And yeah, these were basically repeated. So there were around 120 trials in for each participants, and in blocks of 10 trials. So they were walking around like half an hour, then they took a break, and then they were walking again half an hour. For EEG analysis, we used independent component analysis. Yeah, probably many people here are familiar with the method. ICA basically separates the EEG and temporally independent source signals and allows to separate brain sources and artifact sources, which is especially important in MoBI research because yeah, we have a lot of artifacts during walking. This is the analysis pipeline. So the channel EEG was decomposed using adaptive model ICA, adaptive mixture model ICA. And then for each IC, we estimated IC equivalent current dipole locations. And then we identified and deselected non-brain artifact ICs using dipole location, the scalp map, the temporal evolution, and the spectra. We then clustered the remaining brain ICs across subjects based on the scalp maps, the dipole locations, and the frequency spectra. And to align the heel strikes of over trials and over subjects, we computed single trial spectrograms, and we time warped them to the median step latency across subjects using linear interpolation. And we did this for the 4 heel strikes and the 10 heel strikes following each tempo change. And to look at relative changes in power, we subtracted the pre-shift average log spectrum, so the spectrum before the actual tempo change here. And significant changes to baseline were computed using a bootstrapping method. So before getting to the behavioral results, let's look a second at the event-related spectral perturbation here, because the figure is quite complex. So on top we have the scalp map and the dipole locations of the cluster, of a right frontal cluster. Then we have the distribution of the cues relative to the steps in the upper panel. And then we have the event-related spectral perturbation, and the vertical blue lines are right and left steps. So 0 is basically the step relative to the first cue, indicating a tempo change. And this was always a right step. And then, yeah, we have the left and right steps that were time warped to be aligned over trials and subjects. So let's look at the behavioral results. Here you can see the gray line in the back is the inter-cue interval. So for step lengthening or for step delay perturbations, those inter-cue intervals get longer or get larger, of course, because the queuing gets slower. And for step shortening or step advanced perturbations, inter-cue intervals get shorter because the queuing gets faster, of course. And then the red line is the step delay perturbation, so when participants have to lengthen their steps, and the blue line indicates the interstep intervals for step shortening. The squares are always right steps, and the crosses are always left steps. And what we can see is basically that after 2 steps, participants are always already in line with the new tempo, so they don't take too long to adapt to the new cue tempo. But when we look at the difference between steps and cues, so how close in time participants step to the cues, we can see that it takes them a bit longer to get back to their previous alignment with the cues, so to the alignment before the tempo change. So it takes them around 5 to 6 steps to step really closer to the cues. Let's look at the EEG results. So what we found is basically a beta power increase in temporal cortex here during the adaptation period. So I call it the adaptation period because it's basically the period when most of the adaptation is taking place. So in the first 2 to 3 steps after the tempo change. We found these beta power increases for step delay perturbations, so for step lengthening, and for step advance perturbations for step shortening. And we also found these beta power increases in a frontal central cluster and in a left frontal cluster. And these all showed this beta power increase for both the conditions. However, we found one cluster in the right frontal cortex that showed beta power increase only for step advance perturbations. And this difference is significant. And this beta power increase occurs during the second adaptation step, which is the step where most of the adaptation performance takes place. So this we interpret this as beta power increase related to action stopping or motor inhibition, because it's the location and the frequency band is very similar to button press studies that have shown the stopping of ongoing movement. Here we can see again beta power increase. So this is the time course of beta power in a band from 15 to 20 hertz. And we can see that for all the frontal clusters, there is a beta power increase, but only the right frontal cluster shows a difference between step shortening and step lengthening. And this difference is significant. So the envelopes around the mean show the confidence interval of 3 standard errors. Interestingly, what we also found was a long-lasting mu and beta power decrease in parietal cortex. So there were 2 clusters, one in left parietal cluster and one left parietal cluster and one right parietal cluster. And what we can see is that the activation is contralateral to the brain area. So basically, the left parietal cluster has activations around the right heel strike, and the right parietal cluster has activations around the left heel strike. So if we look at the time course of this desynchronization, it's more similar to the time course of the step Q adaptation. And this might mean that, so there are studies, many studies have shown that the parietal cortex is involved in visual motor integration. So here it could show something like the integration of the cue and the step together. And it could indicate that the parietal cortex is maybe involved in more continuous smooth adaptations that are less conscious. For example, like if we have to walk over an uneven terrain or something like that, which is different to when we have to actually stop and avoid an obstacle. This is the paper, so if you're interested in more details, you can look it up here. So we have shown in the previous analysis that the adjustment of pre-planned steps require response inhibition. We have shown that gait adjustments involve right prefrontal beta power increase, and this has been shown to be a marker of response inhibition in seated button press tasks. So let's go back a second to the video from the beginning. So if you imagine walking through this crowded street, before you actually adapt your step, you first have to recognize when you have to adapt your step and where you have to go. So here, the error-related potentials come into play. Let's look a second before we get to the analysis. Let's look a second at the gait variability. Gait is highly variable, so there can be variability in step width from trial to trial and between steps, and there can be high variability in step length. And there can be also differences in the step length and step width between right and left step. So this is, for example, this is an example of a subject step width in a subject. And we can see that, so the blue histogram is for right steps and the red histogram is for left steps. And we can see that actually there is a high variability in step width over time in the subject. So if we average all the trials, we lose the information on this variability. And that's what often happens in EEG research. So people average the trials and then they do statistics on the average peak latencies, the amplitudes, the average time series. But single trial analysis provide information that is missing if we collapse the data to a mean. And especially in MoBI, this is very important. So because there's a high behavioral variability, and the environment of MoBI experiments usually is not that controlled. So if we average all the trials, we lose a lot of information. So in the previous analysis, I took care of the variability, or I eliminated the variability by time warping the spectrograms over the trials and over subjects. With the error-related potentials, I will look at the single trial variability of error-related potentials and see how it relates to the adaptation steps. Let's look again a second at the experimental paradigm. So you remember, people first walked 10 seconds without cues, then they walked with the cues with preferred cadence, and then the cues got faster or slower, and they had to adapt their steps to the tempo. Well, let's look a bit more in detail how this looks. So basically for step advanced trials, where participants had to shorten the steps, the cue arrived earlier than participants expected. For step delay trials, the cue arrived later than participants expected, so after they had already stepped. So after the actual heel strike. Let's look again at the behavioral results. So here, the red and the blue background are the inter-cue intervals. So you can see again that for step advanced perturbations, the cues get faster, and for step delay perturbations, the cues get slower. And the histograms are the inter-step intervals. So what's important here again is that actually in the first two steps have the main adaptation performance. So after that, with the third steps, participants are already in line with the new tempo of the cues. So what we will look at here are basically these two steps, these two first steps after the change in cueing. In this case, we used the same analysis pipeline as for the previous analysis, but here we clustered the brain ICs across subjects based on the scalp maps, the dipole locations, and the ERPs. So once we had the clusters, we were looking to select the cluster that had the highest contribution to the scalp ERP that we were looking for. So in this case, we were looking for a negative deflection around 250 milliseconds after the change in cueing. So we locked the EEG to the first deviant cue. And for step lengthening, we observed a negative deflection 250 milliseconds after the cue, which is very similar to error-related potentials. And what we found here is that the cluster in posterior medial frontal cortex explains almost the whole whole negative deflection here. This is also what people have previously found in error-related potentials when they were localizing it and when they were looking with fMRI and error processing, that posterior medial frontal cortex is the generator of this error-related potential. So let's look at the average of these negative deflections for step advance and step delay perturbations. So what we found is that there was like a big negative deflection for the step delay perturbations, but a much smaller one for step advance perturbations. Here we call this negative deflection or error-related potential step delay negativity. So what we did then was we stacked all the trials of all the ICs for this cluster. So here you can see there are around 600 trials stacked here. And then we sorted the trials by the second adaptation step. So you can see here that bigger steps are predicted by a larger amplitude of the SDN. So bigger steps here in this case are actually beneficial for better adaptation because participants had to make bigger steps to adapt faster to the new tempo during step lengthening. So a larger SDN actually here predicts a better step adaptation. To quantify this relationship, we computed a multivariate regression using the first and the second adaptation step as the dependent variables and the SDN amplitude as the predictor. And what we can see here, so on the left, these are single trials for 2 subjects. So we computed a multivariate regression for each subject over the single trials. And what it showed was that there is a significant relationship between SDN amplitude and the second adaptation step. So SDN amplitude predicts, bigger SDN amplitude predicts a bigger adaptation step. And the same thing for trial averages. So when we average the trials and we compute the regression over subjects. You can see that I'm not showing the results for step advance perturbations because for those the regression did not get significant, but there was a trend that larger SDNs for step advance perturbations predicted smaller adaptation steps, which also indicates that they predict a more efficient gait adaptation. So this is the paper. If you want to look it up, there are more details about the study. And I also published the data. So this is the data descriptor. It has more information about the experiment and the structure of the data. And the data is freely available online on OpenEuro in the brain imaging format. So I think there was gonna be a talk tomorrow about the format. And yeah, you can just download the data and try out your own analysis if you want. What have we shown here in this analysis? So what we found is a cortical inhibitory network that is involved in gait adaptation. So on the one hand, we found beta power increase in right frontal cortex during the second adaptation step and during step shortening. And this indicates actually that the step shortening requires response inhibition. And we also showed that error-related potentials in single trial predict the second adaptation step. So they basically predict the speed of gait adaptation and the efficiency. But the prefrontal cortex involvement in gait adaptation probably depends on the type of gait adjustment. So we found on the one hand those long-lasting decreases in mu and beta power in the parietal cortex, which probably are responsible for smooth, continuous adjustments of steps. And then on the other hand, we found this prefrontal cortex involvement in the right prefrontal cortex, beta power increase in right prefrontal cortex, and the error potential that showed a direct relationship between brain inhibitory brain measures and adaptation steps. So what does this mean for clinical research, for example? So for example, for Parkinson's, I think that these results may be very interesting because both the error-related potentials and beta oscillations are modulated by dopamine in the brain. And as we know, dopamine is deficient in the brain motor system in Parkinson's. So for error-related potentials, it has been shown that the amplitude of error ERPs is reduced in Parkinson patients and becomes unresponsive to changes in dopamine medication. Single trial dynamics of the SDN in relation to gait task challenges in Parkinson might be used as, for example, a biomarker of gait adaptation deficits. And yeah, it's possible that this reduction of the error-related potentials in Parkinson's might hinder them to recognize obstacles in time and then implement the behavioral changes, for example. Yeah, for beta power, it's a bit complicated because beta oscillations are Parkinson's patients have aberrant synchrony of beta power in the brain motor system. So they have too much of beta power. So one possibility could be that beta power is already saturated, so they cannot increase it more and they cannot implement these stopping of the behavior, or there is too much of a break, for example, and participants just freeze in front of an obstacle. The results could also be used. So I think the next steps would be to investigate these EEG features in Parkinson's or in other neurologic populations. And then if
we would find the same thing. This could be used, for example, for TMS treatments to tune TMS stimulation to the right frontal cortex, for example, using beta power frequencies, or, for example, to use medication that increases cognitive control to treat gait adaptation deficits, or it could also be used to develop neurofeedback training using oscillations, beta power oscillations, for example, or TMS to increase response inhibition in those populations. Yeah, and for healthy participants, I think that that we should look at the processing pathways of these signatures in the brain. So the advantage of these findings is that those signatures have been studied in seated button press tasks, and there are already some processing pathways known in other movements, such as finger button presses. But I think it would be important to compute connectivity, for example, to see how are the processing pathways during gait adaptation. And of course, to do this also in different, like, for example, in in a visual domain, for example, to avoid obstacles that are projected in front of you or something like that, because this experiment is using auditory cues, but this is actually kind of like an unusual task if we think about our everyday life, because we are more tuned to adapt our steps to visual obstacles or things that we can see, because we mostly navigate with our vision. Yeah. Okay. That's it. So thanks to all the SWORD Center, and especially Scott McKay, who supported this analysis, and yeah, who I enjoyed very much to work with on this. Thank you.